Now in the second part of the complement video, we will be reading about the different pathways of the complement activation. We have learned that there are three pathways of the complement activation, that is the classical pathway, the mannose lectin pathway and the alternate pathway. But before reading all of these three pathways, we should know that in all these three pathways there are four common steps of complement activation. The first step is the initiation of the complement activation. The second step is the formation of the C3 convertase. Then the third step is the formation of C5 convertase, and the fourth step is the formation of the membrane attack complex or the MAC. Now we should also know that the final step of all the pathways, that is from C5 till the formation of the membrane attack complex, is similar in all the three pathways. Okay, so from C5 to membrane attack complex, that means the final stages in all the pathways of complement activation are similar to each other. So the variation is only in the upper part of the complement activation. Okay, the variation is only in the upper part of the complement activation pathways. Now we will be reading one by one about the activation of the complement system. First of all, reading about the classical pathways. So we know that the classical pathway begins with the antigen antibody complex. So what happens is that whenever there is a antigen antibody complex, then the C1q, that means we know that the C1 complement protein, the C1 complement proteins consist of three components. It has got three components that which are named as C1q, C1r and C1s, out of which the C1q binds to the FC portion of that antigen antibody complex. So first of all, this C1q, okay, the C1q portion goes and bind to the antigen antibody complex. After binding of the C1q properly with the Fc portion of the antigen antibody complex, then the C1r and S components of the C1q complement protein get activated, and this all happens in the presence of calcium. Okay, these all reaction happens in the presence of calcium. So once this C1rs part of that C1 complement protein get activated, we know that the C1s component of the the C1s component of the C1 complement protein has got some esterase activity. That C1s is very important here, that has got the esterase activity. That C1s component, I mean, due to presence of that C1s component of the C1 complement protein, there is breaking in the C4 complement protein. So once the C1 complement protein gets activated, then there occurs the breaking in the C4 complement protein, and after breaking of the C4 complement protein, there is formation of the C1 C4b complex. Okay, and the C4a component of the C4 complement protein get out of the pathway and get involved in some other functions. Now once there is formation of C1 C4b complex, then this converts the C2 to C2a and C2b. So C2b here, since C2b is the smaller fragment of the C2 complement protein, then here the C2b component get out of the pathway, while the C2a get involved in the formation of a complex, and thereby there occurs the formation of the C1 C4b C2a complex, which is otherwise known as the C3 convertase. So this is the second step in which we are getting this C3 convertase. So once there is formation of this C3 convertase, then by the name itself we get to know that it will convert C3 to C3a and C3b. Now since the C3b is the larger fragment, so that will be involved in the formation of the further complex. So here the next complex that is formed is the C1 C4b C2a and C3b. So this is the this is otherwise known as the C5 convertase, while the C3a get out of the pathway and get involved in some other functions. Now once there is formation of the C5 convertase, then further pathway is similar in all the other pathways also. Okay, the final steps in all the pathways are similar. So we will read the initial portions of all the pathways one by one, and we will see the final steps at the last. Now come to the initial steps in the lectin pathway. So what happens in the lectin pathway is that there is mannose residues on the microbial surface. So once the body sees that there are mannose residues on the microbial surfaces, then we have some lectins in our blood, that is the mannose binding lectins. Those mannose binding lectins go and bind to those mannose residues of the microbial surfaces. And once this happens, once this complex of mbl is formed with the mannose residues on the microbial surfaces, then we have also the mbl associated serine proteases, which are in short called as masp. These are nothing but the serine proteases. They also come and bind to that complex, and thereby they form the mbl masp complex. Now this complex, since this complex has serine protease activity due to presence of masp, so it acts as the acts as an enzyme and breaks down the C4 to C4b and C4a. Now since C4b is a larger component, so C4b is involved in the formation of further complex, and C4a gets out of the pathway and get involved in some other functions. See here, the next complex that we are getting here is the mbl masp and C4b. Okay, now this acts as an enzyme for the C2 complement protein, and then the C2 complement protein is further broken down into C2b and C2a. Now in this complement protein C2, the C2a component or the fragment is bigger than the C2b fragment. So C2a fragment is involved in the formation of the further complex. So see here, the next complex which is formed is the MBL-MASP-C4B-C2A. So here we had C2A, and here there is addition of C2A. Okay, after C4B. Okay, so and C2B get out of the pathway and get involved in some other functions. So this complex now acts as a C3 convertase. So this is the second step in the complement activation. So C3 convertase formation is the second step. So we have got the C3 convertase here, that is MBL, MASP, C4B, C2A. Now this complex breaks the C3 into C3A and C3B. So out of which the C3B fragment get involved in the formation of the complex, while C3A gets out of the pathway since it is a smaller fragment. Okay. Now what we get is the MBL, MASP, C4B, C2A and C3B complex. This is the C3 convertase. So this is the third step. The formation of C5 convertase is the third step. So in the classical pathway also we had got the C5 convertase. Here in the lectin pathway also we have got the C5 convertase. Now let's see the alternate pathway. There also we will see the C5 convertase. So see here what happens in the alternate pathway. The starting point is always the antigens of the pathogens. So here what happens is that the bacterial endotoxins, the yeast cells and the viral antigens and the cobra venoms, they cause the initiation of the alternate pathway. They cause the spontaneous hydrolysis of the C3 protein, C3 complement protein. So C3 here spontaneously gets broken down into C3A and C3B, and once there is formation of the C3b fragment, then there is addition of the factor B. The factor B is nothing but the plasma protein which comes under the properdin system under complement system. So complement system, we know in the first part that complement system was classified into three subgroups, one of which was the properdin system, and the protein which comes under properdin system is nothing but the factor B. So here the factor B is involved, and C3b and capital B, factor B fragment forms a complex, that is the C3b capital B complex. Okay, and after that there is formation, in the presence of factor D, what happens that the factor B gets broken down into B smaller, B a smaller fragment and BB larger fragment. So here this BB larger fragment gets involved this BB larger fragment gets involved in formation of the C3 convertase, okay, while the smaller fragment gets out. The smaller fragment gets out and get involved in some other functions. So here what we get is the C3 convertase as the C3b and capital B small b. This is the our C3 convertase, and this is the second step of the complement activation. Now we are we should get the C5 convertase in the third step of the complement activation. So see here, how do we get the C5 convertase? So once we get the C3b capital B small b complex, then this acts as the C3 convertase. This acts as C3 convertase, okay, this acts as C3 convertase. So this C3 convertase now further again converts the C3 into C3A and C3B. So since C3B is a larger fragment, so that gets involved in the complex formation. So see here, there is formation of C3B capital B small b and then C3B complex. Now this complex acts as the C5 convertase. That is the third step in the complement activation. So in all the pathways we have come till this point that C5 convertase has been formed. Once the C5 convertase is formed, then what happens? Let's see here. Once the C5 convertase is formed, then the C5 is broken down. The C5 complement protein is broken down into C5b and C5a. Now since C5b is larger fragment, so that is involved in further complex formation, while C5a gets out from the pathway and get involved in some other functions. Now see here, the C5b is further associated with C6, C7, C8 and C9, and thereby they form the C5b, C6, C7, C8, C9 complex. This complex is otherwise also called as the membrane attack complex. What does they do? This complex forms a pore on the bacterial surface or the viral surface, and once the pore is formed, there is influx of different types of substances, fluids, etc. into the cell of the bacteria, and there are bursting of the cell and thereby lysis of the bacterial or tumor cell. So this is how the complement system augments the the pathway of the augments the immune response by killing the microorganisms path or the tumor cells or viral cells, etc., or the bacterial cells, etc. So see here, this was the final pathway in the classical and the lectin pathway. Also this was the pathway in this is the final steps in the alternate pathway also. See here, the C5 convertase which was formed, that forms the C5b fragment, and then C5b gets associated with the C6, C7, C8, C9 and forms the membrane attack complex that causes or leads to formation of pores in the microbial surface and thereby lysis of that particular pathogenic cell or the tumor cell. So this is how the different complement pathways lead to the formation of the membrane attack complex and cause the cell lysis and thereby augment the immune response. So these are all the pathways. This can be remembered only if you practice these all pathways 2 to 3 times at your home. Okay.